Hello everyone. First of all, I would like to thank the organizing committee of ICAZ 2020 for providing me this opportunity to share my research with the scientific community. My name is Iqra Sarfraz and I am currently working as PhD scholar at Department of Zoology, Government College University, Faisalabad. Today I will be talking about hasparatin as a promising drug candidate against COVID-19. SARS-CoV-2 is a rapidly spreading new strain of coronavirus which has affected almost all the countries of the world and it received worldwide attention. The lack of specific drugs and vaccines against SARS-CoV-2 is a major hurdle towards the prevention and treatment of COVID-19. Thus, there is an urgent need to boost up research for the development of effective therapeutics against COVID-19. The crystallized form of SARS-CoV-2 main protease was demonstrated by a Chinese researcher and which emerged as a novel therapeutic drug target for the development of anti-COVID-19 drugs. This study was conducted to evaluate the efficacy of customized dietary nutrients-based virtual library against COVID-19 virus main protease by molecular docking study. But why main protease? Main protease is a key protein required for the proteolytic cleavage of the viral polypeptide in order to release the functional proteins such as endoribonuclease, exoribonuclease, and RNA polymerase. The functional significance of main protease, as well as its absence of its close homologues in humans, identify or demonstrate main protease as an attractive target for the antiviral drug discovery. That's why we were focusing on the main protease. And so what was our screening strategy? Our screening strategy include molecular docking investigations using molecular virtual docker 7 to analyze the binding interactions of these dietary nutrients based virtual library against COVID-19 virus main protease. The structure of COVID-19 virus main protease was assessed from protein data bank, and this all the natural compounds in the library was docked against the COVID-19 main protease structure. And according to our obtained results, what we obtained results, hesperidin, rutin, diosmin, and apin was found to be the most effective against COVID-19 virus main protease. However, among all these potent hits that we obtained after docking of these compounds against COVID-19 virus main protease, we found that hesperidin exhibited the highest binding energy at the active site of COVID-19 with the mole dog score of -187. And hesperidin formed strong hydrogen bond interactions with number of amino acid residues of the SARS-CoV-2 main protease. And one of the previous study by Lin et al. Has also reported the incredible in vitro activity of hasparatin, which is a primary metabolite of hesperidin against SARS-CoV main protease, which also further justify our results. So, keeping in view of the incredible potential of hesperidin against COVID-19, one of the most important concerns that need to be considered is that whether hesperidin will be bioavailable after oral administration, and what about its toxicity and its safety profiling. So, luckily, we have some overlapping results from our previous studies, and based on our results from Park the Researchers Mobility Grant to explore the dietary nutrients as leads for anti-cancer drug development, luckily we have also identified hesperidin as an anti-cancer agent targeting tumor metabolism. And our data and results from the previous studies clearly show that drinking 1 liter of orange juice provide 444 mg per liter of hesperidin that reach a peak plasma concentration of 1.3 micromolar in 5 to 7 hours. And this study clearly demonstrate that hesperidin possess a very good oral bioavailability. And another vital concern while developing therapeutics is the safety and toxicity profiling of the drug candidates. So interestingly, the toxicity of hesperidin is very low. It has a lethal dose at more than 2 gram per kg body weight of the experimental animal. And even more interestingly, hesperidin do not possess any toxicity, hepatotoxicity, and nephrotoxicity at the dose of 1000 mg per kg of the body weight of the organism. This data clearly demonstrates that hesperidin possesses all the drug-like properties, and it could emerge as a promising drug candidate for the development of drugs against COVID-19. So this study has unveiled the potential of dietary compound hesperidin as a cheaper and safer drug candidate against COVID-19. However, large-scale clinical trials are urgently recommended to prove the efficacy and safety of this drug candidate in COVID-19 patients. And at the end, I would like to acknowledge the HEC Park Berg Researchers Mobility Grant for providing funding to support the second part of this study. And at the end, I would like to thank you all for your patience.